generous introduction. And it's a great pleasure to be here, and particularly it's an honor to be hosted by the Early Career Committee. So basically what I would be talking about is the vasculature and how it impacts the tissue regeneration and aging. And of course, as my lab is interested in bone, but when we talk about vasculature, we have to always compare with how it is differing with the other organs. So I will be giving a more holistic approach where how the vasculature changes with age in different tissues and how it regenerates across different tissues, including bone. So basically, what my lab, I have done my postdoc in Germany, and when my lab moved to Oxford, for what we are interested is how the vascular diversity changes over time and how it impacts the tissue regeneration. Particularly, we are interested in identifying the therapeutic target to treat the age-associated diseases. So we know vasculature is important because the blood vessels supply oxygen and nutrients to the tissues. But in addition to their role as the transport network, the vasculature is important. For example, the blood vessels play a critical role during organ development, and they are critical for the tissue repair. But also these lymphatic vessels are known to play important role during the inflammation. So these vessels are just not tubes or the transport system. And the endothelium, which is the endothelial cells, which are the cells which lines the vasculature, and these pericytes can engage into the signaling with the neighboring cells. I just want to have this, please. Okay. So, and this could be I just want to switch on the laser. Just a second. Okay. So yeah, so the endothelium and pericytes, these can actually engage into the signaling with the neighboring cells, and these neighboring cells could be a stem cell, immune cell, or the cancer cell. And this is the kind of interactions which we are looking at, because when the vasculature interacts in a paracrine or justacrine manner with these neighboring cells, they can govern the fate and behavior of these cells. And particularly, what my lab is interested is also in understanding how these interactions change with age. So basically, what are the changes in this age-dependent changes in these interactions in the paracrine and justacrine signaling with the age, and how the vasculature itself change with age, and how then this impacts the tissue regeneration, because we know as we age, the capacity of the tissues to regenerate declines. So this is where we are asking is how the aging impacts tissue regeneration. And when we talk about the aging of vasculature, it is actually if it's like in the 16th century, this is a British surgeon, Thomas Sydenham, who quoted that a man is as old as his arteries, meaning that as we age, the arteries stiffen. So, but until recently, not much has been known about how the capillaries age, how the vasculature in different organs age, and how are the changes with the lymphatic vessels, for example. And I am interested in bone because we know that bone hosts these blood and the blood stem cells, which can actually give rise to the blood and immune cells. And here, for the blood stem cells, the vasculature is known to be the niche for these cells. So what we did was we asked how the vasculature changed over time across different organs. And towards this, what we did was we did a tissue imaging approach across many different organs listed here. And what we found that as we age, there is a decline in the vascular density. Here I'm showing you the example of the spleen and organs which host these, which also have the immune cells. And we see that there is a decline of the vasculature. Here you can see this by the staining for endomucin. So red shows the staining for endomucin, And endomucin is a marker for the capillaries. And you can see that the aged tissues shows decline of the vessel density, and we can quantify that. And we can see this decline of the vascular density with many other markers, meaning that it's not the loss of endomucin expression, but basically with many other endothelial markers, this can be confirmed there is a loss of vessel density with aging. And all this data, which we have not only done for the endothelial, but pericytes, fibroblast, and other matrix marker is available as on the Omero server as the 3D tissue maps. So basically, what to summarize this, what we have found is that as we age, there is a loss of vessels, but there is also this loss of surrounding pericytes. But more interestingly, what we found in both the mouse tissues and the human donors is that there is an accumulation of fibroblast with aging. So while there is a loss of vessel density, loss of pericytes, there is an accumulation of the fibroblast. So we asked why these pericytes declined with age. And towards this, what we did was we did a lineage tracing approach. And for the lineage tracing, what we used was PDGFR receptor beta. So it's platelet-derived growth factor receptor beta, and this is a marker for the pericytes. And when we do the lineage tracing, here with the tomato label, you can see all the pericytes. So these are the vessels, and the FSP, which is a fibroblast-specific protein 1, is a marker for the fibroblast. And when you compare these young tissues versus the aged tissues, what we find that during the aging, these tomato expressing pericytes start expressing the fibroblast marker, which is the fibroblast specific protein 1, meaning that with aging, there is a differentiation of the differentiation of the pericytes to the fibroblast. And this is why we see that with aging, there is an accumulation of the fibroblast, both in human and the mouse tissues. So we asked, what happens during the diseases such as fibrosis or arthritis in these diseases, because fibroblasts are known to be the pathological drivers of these diseases. And so we asked whether pericytes can contribute to fibroblast in such age-associated diseases, and the answer is yes, because if we do the lineage tracing using, again, the PDGFR beta, which is the marker for the pericytes, and if we look at the expression of the fibroblast-specific protein 1, and in this experimental arthritis model, we can see that there is a co-expression. These tomato-positive pericytes are expressing the fibroblast marker, which is FSP1, meaning that during these diseases such as arthritis or fibrosis, I'm just showing the example of arthritis here, but we have also looked at the fibrosis. Pericytes can differentiate into the fibroblast, meaning that in this situation, we can target the vasculature to inhibit the progression of such diseases. So far, what I have shown you that the pericytes can differentiate into the fibroblast, not only during aging, but during the age-associated diseases as arthritis and fibrosis, thereby vasculature can be targeted during these conditions. But also what we observed when we looked at the mouse, we looked at the mice at the different ages, what we saw that these changes we find in these tissues, such as the loss of pericyte or loss of vessel density, is coming much earlier than the known hallmarks of cellular aging, such as mitochondrial dysfunction or cellular senescence. So what we did, we asked what happens upon the vascular loss in the young mice. And to test this, we interfered with the VEGF-R2 signaling in the young mice. So basically, VEGF-R2 is vascular endothelial growth factor receptor 2, and this is important for the proliferation and the maintenance of the vasculature. And we specifically deleted VEGF-R2 in the endothelial cells using weak ethylene pre-RT2. Here we used the 8 to 10 weeks mice. These were the young mice. And what we see, as expected, if VEGF-R2 is deleted in the endothelial cells across many tissues, there is a vascular loss. And interestingly, this vascular loss is sufficient to drive the cellular hallmarks of the aging, such as mitochondrial dysfunction or cellular senescence or stem cell exhaustion. And here, what I'm talking about is these changes is not only in the endothelial cells, but we see these changes in the surrounding cells, meaning that the changes in this vessel density is sufficient to induce the tissue aging. So we then looked at the molecular differences between the young and the aged endothelial cells. And when we looked at the the differences between the young and aged endothelial cells, this we did by the bulk RNA sequencing of the young and the aged endothelial cells. And here, what we noticed is that in the aged endothelial cells, the HIF, notch, hippo, and Wnt signaling decline. And this was, specifically the HIP and the notch signaling is known to be critical for the vascular maintenance and the endothelial cell proliferation. And we see that as the vessels age, as the tissues age, the endothelial cells sorted from the aged mice have decline in these pathways which are critical for angiogenesis and vascular maintenance across many of these tissues listed here. And what we find is, like I have been talking about all these tissues here, that there is a loss of vessel density, there is a loss of vessel densities, but other tissues which can retain the vessels include skin, gut, and the bone. And we know that these tissues have higher these organs, these tissues have higher regenerative capacity as compared to the tissues such as heart or kidney. So basically, we compared the molecular differences between the tissues which can retain the vasculature versus the tissues which cannot retain the vasculature. And what we find is that the tissues which lose the vasculature with the age, which is this set of the tissues, compared to the ones which are able to retain the vessels with the age, is the difference is that these endothelial cells are highly pro-inflammatory. Here we see that there is expression of many of the cytokines such as IL-6, IL-1 beta. So, compared to the tissues which can retain the vasculature with age, the vessel density with age. So, coming to bones, and we know what we have previously done is that we have looked at, and we know that the vascular the vessel density, the vasculature within the bone is highly heterogeneous, and we could actually use different markers such as CD31 and endomucin to characterize different subset of blood vessels in bone. Particularly, we have identified this type H vessels, which are CD31 and endomucin high, and which are present near the growth plate of the bones. And we see that actually with aging, there is a decline of these type H vessels, which are high for endomucin and CD31, but the vascular density do not decline with age in the bone. So for this, what we have done is basically looked at the thick sections of the bone, but this is not sufficient. If we have to look at the lymphatic vessels, for example, or if we have to look at the if we have to analyze the larger vessels such as arteries, for which we have to image the intact bone. And the problem here is that with the imaging of the intact bones or intact calcified tissues, is we know most of these methods basically depend on the reporter mice. The immunostaining is challenging because there is a decalcification step which affect the antigenicity. And then what we also other issues with these kind of imaging is the data analysis and the high amount of data which is generated. So what we did is we have developed this workflow where we have basically we can look at the whole bone, we can clear it, but also most importantly, we can do the whole bone immunostaining. And this is very critical because we do not depend on the use of the reporter mice, and this allows us to look at the vasculature, but even the rare cell types like the hematopoietic stem cells in the bone. So basically we are clearing the bones, and before that we are doing the immunostaining. And towards this, we use we have used ultra microscope too most of the time, but most recently we are using the Blaze, which is also from the LaVision Miltony Biotech, and we have these analysis pipeline, and we can do the quantification. So these are some of the images which we have generated. So basically, if you see, this is the tibia, which is after clearing, and this is a murine tibia, and we can see that with the stainings for the vascular markers, that basically the whole intact tibia, we can look at after imaging the vasculature. So as I have shown you the example of the murine bone tibia, here are the examples of the other bones. So basically, we can look at any mouse bones, but most importantly, we can also use the human bone biopsies. We can apply this method for the high resolution imaging of the human bone biopsies, and based on how the imaging is performed, we can do the analysis. As I said, as my lab is more vascular focused, we are looking at the vessels, but we can look at any other cell type by immunostaining with these antibodies. And here, for example, here is the endomucin staining, which would show all the capillaries, and then there is alpha smooth muscle actin staining. Yeah, so this is a knee joint, and you can see the arteries in this knee joint with the cyan color. So basically, this imaging method can be applied across any tissue, any of the calcified tissue, and the imaging can be performed after the immunostaining. So basically, coming back to the question of the vasculature, so the current dogma is that the lymphatic vessels are absent in bones. So, but with this method which we have developed, we asked whether this is true or not. So we tested this dogma, and what we found, as we see here, with the immunostaining for the lymph-1, which is a marker for the lymphatic vessels, and it's shown here in red, and we can see that the staining for the lymphatic vessel marker shows that the lymphatic vessels are present in the murine bone tibia. And we can also see, so it's known that the lymph-1 can also stain a subset of macrophages, and we can see by the staining of the macrophage markers that they like F480, that these vessels are not positive for the macrophage markers. That means they are the lymphatic vessels, but not the macrophages surrounding the vessels. And this is how we can look at this. And if you see, this is the whole bone, whole cleared tibia. And we can see here that there is these there is this lymphatic vessels in the bone, which is in the bone marrow. And here, which is this is a higher magnification version. And we can differentiate this with the Lyve1 GFP reporter mice versus immunostaining. We have also done the immunostaining here with CD102. And now CD102 marks the vascular, the blood vessels, but it is not marking the lymphatic vessels. So if you have a look at this video, so the red shows the blood vessels, and these are the lymphatic vessels. So these lymphatic vessels in the healthy bones are mostly concentrated in the cortical region of the bone. And this is the bone marrow. And here is the bone marrow. There is this central lymphatic vessel going there. And then you see this, basically they are situated. They are more localized within the cortical bone in the healthy bones, but not in the bone marrow. And we can detect these lymphatic vessels across basically many other bones, but also human biopsies, as it can be seen here, human bone biopsies. And what we did was that we asked, what is the function of these lymphatic vessels in bone? And towards this, what we did was we looked at different conditions. And so what we observed that during the injury or stress condition, and here is the radiation stress, we see radiation stress, we see that these lymphatic vessels expand dramatically. And here you can see this by the Lyve1 immunostaining. So to understand what is the function of these lymphatic vessels during the injury and stress situation, and why do they expand during the injury, what we did was that we genetically depleted these vessels, and also we use the pharmacological inhibitors of the lymph angiogenesis. And by both the approach, what we can see is that if we ablate these vessels, and if we ablate lymph angiogenesis, if it is inhibited, then we see that the bone regeneration is impacted. And so basically here, for this particular experiment, we have used PROX1 CRE-RT2, because PROX1 is a very specific marker for the lymphatic endothelial cells, and its presence is sufficient to differentiate the endothelial progenitors into the lymphatic endothelial cells. So we used this marker. We used the PROX1 CRE-RT2, and we used the diphtheria toxin antigen, and we have, and as you can see here in these CRE-positive mice, there is less bone mass and less bone density after the radiation stress. So we know that in bone, these the bone cells are coming from these skeletal progenitors, the skeletal stem cells, which can give rise to the adipocytes and chondrocytes and the osteoblasts. So what we did was we asked, which of the mesenchymal stem cells or the pericytes are involved during this differentiation, during this pericyte to fire, during this during this regeneration of the bone. So what we did here, so specifically when we were looking at various markers for the mesenchymal stem cells and the pericytes in the bone, what came up was this MYH11, which is a myosin heavy chain, and it is basically expressed in the smooth muscle actin positive pericytes. And this is very interesting because these smooth muscle actin pericytes, these are present around the mature arteries in the bone, and you can see this. These are quite restricted to the bone marrow. But interestingly, if the mice are irradiated or during the stress or injury condition, there is a rapid expansion of these MYH11 positive pericytes, which are mature pericytes, into the bone. And this is quite striking because these pericytes are mature. Even for a pericytes, it's quite down the lineage. So not only we see that they expand during these mature pericytes are not only expanding during the radiation or injury condition, but they can also give rise to the different subsets of they can differentiate into the chondrocytes, adipocytes, and the osteoblast in the bone. And this is why we looked at the we depleted these cells, and we can see that if we deplete these cells, we can do the genetic ablation. There is a delay in the regeneration of the bone after the radiation. And this was very interesting because what we can see is that this expansion of the MYH11 positive pericytes in bone is restricted only to these tissues, because after the radiation, we cannot see this expansion of the pericytes in other tissues as listed here. So basically, this is something very restricted to bone. So then we asked, do these mature pericytes give rise to these bone cells during the bone during the development? And the answer is no, because during the development, these mature pericytes are only restricted to the vessels here, which we can see, which is in the bone marrow, and they are not giving rise to any of the bone cells during the developmental situation. So basically, dysfunction of these mature pericytes to differentiate into the different cell types is restricted only during the stress and injury conditions, which may also explain why bone have a higher regeneration capacity compared to other tissues, because the bone harbors many of these stromal or mesenchymal cell types which are expanding compared to these tissues where we do not see the expansion after the radiation or injury stress. So what we did next was we blocked angiogenesis and to see what is happening to these cells. And if we block lymph angiogenesis, again, what we see is that these pericytes are not able, if we block the lymph angiogenesis, these mature pericytes cannot give rise to the bone. So it is dependent on the lymph angiogenesis. And how it is dependent on the lymph angiogenesis is that because during the injury situation, there is an upregulation of IL-6, and IL-6 is a known driver for lymph angiogenesis. And this proliferating endothelial cells then starts expressing, when there is a lymph angiogenesis, they start expressing CXCL12. This MYH11 positive pericytes also express CXCR4, and they can expand in this situation, giving rise to the other bone cell types. So basically, this CXCL12 secreted from the lymphatic vessels is critical for the differentiation of these MYH11 positive CXCR4 positive pericytes into the bone cells and for the tissue to regenerate. So what I have shown so far is that imaging of these intact skeletal elements, we can find that there are lymphatic vessels in the bone, and these lymphatic vessels, drive the expansion of the mature pericytes, which are Myh11 positive, CXCR4 positive pericytes, which drive the tissue regeneration. And also what we have seen is that the aging impacts this capacity of the the proliferation of the lymphatic endothelial cells, because what we see that after aging, there is during aging, there is intrinsic changes within the lymphatic endothelium, and they do not proliferate. In the interest of time, I have not shown you the data, but we have seen that in the aged mice, there is no proliferation of the lymphatic endothelial cells after the radiation stress. Due to the intrinsic aging of the lymphatic endothelial cells. And basically what we see is that despite the IL-6 expression in aged lymphatic endothelial cells do not proliferate, and that which also leads to the which also impacts the regeneration of the aged bones. Further, now, with this discovery of the lymphatic vessels in the bone, we are currently, these are some of the experiments which are ongoing, because the presence of these vessels in the bone have immediate impact on understanding how these vessels will impact the cancer cells in the bone, particularly during the bone metastasis, and how it impact the acute and the chronic inflammation and the production of the immune cells, but how they respond during the infection. So what we are looking at, how these lymphatic vessels are impacting the cancer cells in bone, and how this changes in the aging, and also how these lymphatic vessels impact the production and the response of the immune cells in the young versus aged mice. And this is what we are currently looking at, how the vascular niches, the lymphatic niches, and their interaction with the immune cells in bone, and how it changes during the aging using the influenza and the malaria model. So these are some of the current experiments which is going on in the lab. And with this, I would like to thank some of my team members. So these are mostly the current team members, but there are a lot more which have been there during the last 200. I started my lab in 2017. So yeah, this is all the work I have been presenting coming from the lab. And I would also like to thank the funders, and thank you very much for your attention, and I'm open for the questions.
Thank you very much, Anjali. Very, very nice talk. So if